I will wait for people to.
Okay.
Sorry, I can't see your presentation now.
Oh, you cannot?
No, it's black.
Okay. Oh,
When it's not on full screen, I can see it, but when you go full screen, it shows a black screen.
Okay, that's a problem, I guess. Let me see what I can do about it. Just a second. Oops, okay. Okay. What can I do with this? All right, let me try one thing.
Okay.
It's fine.
I think some people have joined, so I will give some introductions.
Yeah, sure.
So thank you to the people that have joined. Hello, everyone. Now it's working. So, first of all, thank you for joining. I would like to remind you that if you want to know about the upcoming webinars or how you can present, you can follow us on social media. Today, Pranav Khade will be presenting. He is a PhD student from Iowa State University, and he will talk about the use of alpha tapes to characterize protein flexibility. So now I leave you with him. Thank you so much for doing this presentation.
All right. First of all, thank you so much for the wonderful introduction and giving me this opportunity to present my work. So I'll jump right into it. If you don't recognize some or many words from the topic, don't worry, I'll be explaining them all. So let me start with basics. First of all, what is protein packing? So basically, in order to protein to work, it has to take certain shape. And that shape is possible because of the spatial conformation of the amino acid with respect to one another. In other words, how protein is packed. So from the name, you can see that in order for the protein to work, some parts of the areas needs to be densely packed and some parts of the protein are not so densely packed. So we are going to study this property or this characteristic of the protein and use it to predict the dynamics. How is because we know that like for any other object, if something is densely packed, it has fewer degrees of freedom to move around. Whereas if something is not so densely packed, it is more flexible. It has more degrees of freedom to move around. So we are going to exploit this property in order to see what effect it has on dynamics and if we can predict anything from it. So how it is multi-scale is you'll see later in the presentation, but just for your information, if we apply this technique which I'm going to explain on all the atoms of the protein, we will be able to see the local dynamics, or what are the changes happening on the local level, local time scale. Whereas if we take only the backbone atom of the protein, which is kind of co-screening the structure, we can make the inferences about the global dynamics of the protein. So since this technique is completely based on protein packing, we have to see indeed if it has any effects on the protein dynamics. Now on this graph, I want you to pay attention to the second bar graph, which is labeled as buried. You can see my cursor, right? All right, I assume you see my cursor. So this buried residue is basically the residues which are very densely packed. Okay, and you can see if the mutation occurs in the residue which is buried deep inside the protein, aka very densely packed, it twice as likely to result into a disease. So since there is a mutation, there is disturbance in the structure. Since disturbance in the structure, disturbance in the dynamics, and therefore the function, and therefore the disease. So protein packing is indeed very important for the function of the protein. And in our lab, we also confirmed from our analysis of compliance, which is another way to model the protein packing, which I'm not going to discuss today, but we have confirmed this, that protein packing is important for the dynamics of the protein. So with this information, how to model this important property is very simple. Now, in this diagram, if we start with random 3D points in space like this, and we tune a parameter called as alpha, let's consider it is just a parameter right now, do not think of anything else. So there lies an alpha value which is very perfectly describes the surface of this set of points. And from this surface, we can clearly infer that what is the torso and what is the head and what is the neck region in this diagram. And we know that neck is most flexible part, because it is not so densely packed. And we apply exactly same principle into the protein. So we start with some alpha value, which is like this, and then we tune in the alpha value in order to get an alpha value which can separate the domain 1 from domain 2 and the region which connects two domains, which is a hinge. Now, this is a very trivial case, and hinges are not very obvious all the time, especially if the protein is in the closed conformation. So for that, we have built this package called as packing and motion analysis, or PAC-MAN, and I will show you how it works. But before that, I'll be explaining what are the Delaunay tessellations, because they are central to the concept of alpha shape and therefore the algorithm of this tool. So this is let's consider if we have set of points, let's say these points you see here, and if you provide these points and ask for an Delaunay tessellations, you will get figure something like this. Now, how is this figure generated? That's the question. So in order to answer that, I want you to focus on these 3 points. You see, in 2D space, if you provide 3 set of points, it can form only one triangle, and it's unique. There is no other solution to it. It forms only one and one triangle. So what is the property of this triangle? You can see that there is a circumcircle, aka the circle which has all 3 points on its surface of the triangle. This circumcircle exists such a way that there is no other point inside that circle. You see, there is no other, this is completely hollow circle. And same goes for all the other triangles you see in this diagram. There is no point inside the circumcircle which is formed by the 3 points. And that is how the Delaunay tessellation work. That is how the Delaunay tessellations are derived. you have set of triangles which form a circumcircle, which do not have any other point inside the circle formed by uh, the circumcircle. And this is the concept we apply in 3D. So just like we have triangles in 2 dimensions, in 3 dimension, triangles become tessellations. And in 3 dimension, this circumcircle becomes circumsphere. So all the 4 points of the tessellation lie on the surface of the sphere. Those are called the Delaunay tessellations. And those spheres are always empty. And this is the key concept. Now we will be learning how we will be getting the alpha shape from this Delaunay tessellation. So I want you to remember that this circumcircle exists. Okay. Now I will be explaining how the algorithm works. So first we start with the protein, and we extract the, like it can be in any conformation, open conformation or closed conformation. And we extract the backbone atoms of the protein. And once we do that, what we do is we build the Delaunay tessellations by the process I just explained you. And once we form the Delaunay tessellations, we have different tessellations. That is the group of 4 points, and each of them have this circumsphere. Okay. Now look here. Let's say for a particular tessellations, we go through all the tessellations, and for a particular tessellations, we have points A, B, C, and D, and we have a sphere, a circumsphere, and that circumsphere has certain radius. Okay. Now this is where the alpha parameters comes in. So alpha value is a parameter which tells us if we want to keep this set of 4 points inside a alpha shape or not. Okay. So if let's say the alpha value is 10 and the radius of the circumsphere is 12, we do not keep those set of 4 points in the alpha shape. We just remove them from Delaunay tessellation and just keep the ones which have radius less than 10. And that's basically how we go from Delaunay tessellations to alpha shape. And once we get the alpha shape, it has all the information about the packing of the protein, which areas of the protein are densely packed, which areas of the protein are not so densely packed. Okay. And as we convert this alpha shape into an undirected graph, and once where we connect all the 4 points inside the tessellation to each other, like so A will be connected to B, C, and D, and so on. And once we get this undirected graph, like any other undirected graph, it has centrality properties. Okay, so one of them is called eccentricity. Now eccentricity is very simple. Basically, it tells you what is the minimum distance from any point in the graph to any other point in the graph. And you can guess that if certain point lies inside the hinge, it will have the shortest distance to both the domains, you see. And if certain point lies in the domain, it will take long distance to go to the other domain. So on average, the points in the hinge regions in the graph will have lowest eccentricity. So that's what we do. We consider that the points with lowest eccentricity are the hinges. That's our assumption. Now, these are just geometrically obtained hinges. We want to confirm if they make sense in terms of dynamics. Now, for that, we use a property we obtain from crystal structure, which is B factor. They are often called as Debye-Wheeler factors or temperature factors. So those of you who don't know what it is basically it can be seen as the standard deviation of particular atom, standard deviation of the displacement of particular atom from its mean position. So more it has a standard deviation, or more the B factor is, more flexible it is, more it is moving from the mean position. And we take the distribution of the B factors in hinge region and we compare it with the non-hinge region using permutation test. Now, this permutation test is like a stochastic test. We do this 10,000 times with random combination from hinges and non-hinges, and we get a p-value from that test. If that p-value is less than 0.05, We consider it as a geometrically obtained and statistically verified or dynamically verified hinge. And that's basically it. That's how the algorithm works. We start with a protein in any conformation, and we go on with this algorithm. And in the end, we have a hinge region obtained from PAC-MAN. Now, we tested this method on multiple conformation. I'll be just running through it, and all these examples are manually curated from the literature, so we know that these are the hinge regions. And important thing to note here is that you see this is the protein is in two conformation, but it is the same protein. So, and the hinges predicted in both the conformations are overlapping. So important thing is, given any structure in any conformation, you can predict which part of the protein is going to be flexible and which are going to be allow protein to move or to act as a hinge. And that is a very important information to have. And this hinge was curated from the literature. We verified it from the literature. Same for the RNA polymerase hinge, we verified for the calmodulin, which is a very famous example. Another thing is there can be proteins which can have multiple hinges. As in the HIV protease, there are multiple hinges which allow the hinge to be flexible in order to open the flaps you see on the top. And all these examples are also verified from the literature. Now, sometimes there are not many conformation of the same protein are available. So we decided to apply this algorithm on the protein which do not have multiple conformation. And specifically on the viruses, where knowing the important region can be very helpful, as we know the virus we are dealing with now. So I can go through all the example. I don't think I have enough time, but I want to note this particular example, which is very significant in my opinion. So this is the red region you see here is the loop in the NS3 helicase in Zika virus. If you disable this loop, this protein completely stops. And if this protein stops, the replication of the virus stops. And this is annotated in the literature. And we could correctly predict the hinges which are surrounding this exact region. So if you have information from the PAC-MAN like this, that if particular region is very flexible, you can have the idea about the functioning of the protein and potentially stop its action. I'll be talking about how can we use this information later. But before that, even if you don't do this kind of complicated stuff, even if you have a protein and you run the PAC-MAN analysis on it, you get a general idea how its mechanism can be. For example, you see on your right, you have annexin protein. So you can infer just by predicting the hinges that a red domain is connecting blue and green domain, and they have like leg-like motion in them. So you can get the overall picture of the dynamics of that protein. And same goes for the calmodulin. This blue part comes away from the red part. And we could get this study published where we studied 167 different proteins in open and closed conformation, and we could verify the hinges in all of them. And yeah, this is a very interesting study where we predicted the hinges in the structure. We provided this information of the hinges to the elastic network model. This is NOLB model, by the way. And once we provide this information to the elastic network model, we get this motion, okay? And Dr. Zabotina, who we are collaborating with, wanted to see if we can control the active site or control the activity of the active site in any means. And you can see that because of this flap motion on the top, it has changing specificity depending on the position it is at that current time. So we found, because of this motion and hinges, we could find this spot at the back of the protein, which is a potential effector site. If we block this effector site, this motion which we are seeing, the flap you're seeing, will be restricted, and therefore the specificity of that protein or the active site will be restricted. So this is direct allosteric effect you can infer from the hinges you're getting. So yeah, and again, to reiterate, what is significant is you don't need multiple conformations to predict how the dynamics of the protein will be. You just need single conformation, and whatever hinge you will be getting is statistically robust because it will be obtained from 10,000 iterations of the permutation test. And again, it will be important to predict its mechanism, opening and closing, and potentially effector binding sites. This is ongoing project, and this is under development. We are adding a lot of things besides PAC-MAN into this project. And right now, what we are doing with this information is we are trying to implement the flexible docking. The motions you see, we are trying to implement the docking inside it. We already talking to some people in Kansas University about that. Another thing which I want to do is we want to build a database of all protein hinges. So main goal to do that is so that once you have data for all the hinges, we can profile them. And once you have the new protein, instead of running, before running the molecular dynamics, you can search it against the database, what kind of hinges it has, what kind of potential motion it might have, and so on. So you will have more idea about its global motion before running you will know what to expect from the MD analysis. And also, we are building a new elastic network model based on the hinges. We are collaborating with Politecnico di Torino for that. And this is just a glance of the method which we are developing, but I don't think I have enough time to talk about that. And yeah, I have to thank all the members of the Jernigan Lab for all this study. They are very wonderful group of people, and research IT, all the grants, and most importantly, thank you ISCB Student Council for giving me this opportunity to present my work. Thank you very much.
Thank you, Pranav.
Yeah.
This was very interesting. Now we have to wait a bit for questions. The streaming has a small delay, so we can wait a few seconds to see if anyone has any questions. And I would like to hear a bit more about the flexible docking.
Okay.
I worked a while ago with docking, and I'm not an expert, but it will be how will it work, and what advantages will it have to the traditional method that is used now?
Right. So basically, there is already something called as flexible docking, but it requires huge amount of computational. You can see my screen, right?
No, it's blank.
Okay. Okay, so you could see the motion of the protein which I showed you, which is opening and closing over there, right? Mm-hmm. So we can get those kind of motion from the elastic network model and hinge information we have having. So these are really computational. You just need like matter of seconds to get those motions. And which makes what we can do is we can get different instances of those motions, and we can try to dock the molecules on each of those instances. Like instead of docking it on the solid protein, or introducing it to molecular dynamics, which takes a lot of time, which is effective, but it takes a lot of time. So this is a bit like cheap way to do docking, a cheap way to do flexible docking.
Okay.
Right.
Now we have some questions.
Okay, all right.
So, can you see them, or
I can see them complementary to your?
Okay.
Something completely.
What does the study of proteins by crystallography bring? Is it complementary to your approach, or something completely different?
Well, From the crystallography data, what we are doing is we are using the B factors from the crystallography analysis, and they are like central to the statistical test we are doing, which is permutation test. So that's how we are using the crystallography data. It is not something different. It is part of the algorithm. And so I don't think it is bringing anything different. It is like part of the study. And the second question is, in addition to flexibility, you have any information on mutations or the replacement of amino acid by another? Oh, that's a really nice question. So what we are doing is in the new elastic network model we are building, we are specifying the masses of the amino acids. Okay, so we provide, if certain amino acid is heavy or hydrophobic, it will be included in the elastic network model which we are developing. So yes, eventually we will be able to see the effect of the mutations or replacement of any amino acid by any other amino acid. And we are also planning to include many other properties such as Kidera factors and hydrophobicity and so on. Okay, we have new question. Similar to previous.
Do you analyze the dynamics of proteins from closely related species? Are there differences in flexibility even though the sequence difference is very small?
Okay, that's really good question. Actually, currently the elastic network model do not have this sensitivity which many of the molecular dynamics methods we have. Just because of like single atom replacement, it can have different effect. But we are since this elastic network model is very coarse grained, I do not think we get very useful information from closely related species which have like one or two uh, point mutations. But we are planning, as I said before, we are planning a new elastic network model which potentially can do that. We still have to test it, but we are planning to do that. Right.
So it seems there are no more questions.
Okay.
We can wrap up. Thank you very much for this very interesting talk.
All right.
And next week on Thursday, we have another webinar. So be on the look for the information on our social networks.
Okay.
Thank you, everyone.
Thank you.
Bye.